Ayla, would you please do this? Ayla's a HASI, one of our HASI volunteers. If you just press, if you just set our change.
Okay.
That's what you change in there, okay? That's all. Okay, thanks a million. Right.
Okay, well, I have a little summary of the talk because I always think it's very nice to see where I'm going to go before we start, before we get into the middle of it. So I'm going to go through a few aspects. Firstly, it's about Epstein-Barr virus. And back in the 80s, Epstein-Barr virus was recognized as a significant contributory factor to ME/CFS. But we lost our way a little bit in the following 30, 40 years. And now it's come back from different sources supporting the fact that EBV reactivation is very important. So that's what the talk is going to be about. I'm going to consider different aspects of ME/CFS. For example, the heterogeneity, the fact that this is probably an autoimmune disease, maybe a prodrome to development of other autoimmune diseases, that psychological stress is very important in different aspects of virus infection and in ME/CFS. I'm going to go through a bit about Epstein-Barr virus infection, and then Epstein-Barr virus induced ME/CFS, markers of EBV reactivation, and possible treatments in the future for Epstein-Barr induced ME/CFS. Next, please. So, heterogeneity of this disease. There are a variety of studies that have shown that the disease is heterogeneous. And by that I mean that there may be different subtypes. But it has been very difficult to actually define them because, as we know, ME/CFS consists of multiple illnesses or multiple clinical syndromes which overlap. And so it's very difficult to define subtypes. So these are clinical studies here. And lastly is our gene expression study, which identified 7 subtypes based on the expression of these genes. Next, please. But I think it helps with a disease as complex as this to consider what probably accounts for most cases. And it can be considered to be a flu-like illness triggered by virus infection and mediated by the immune response. And thank you. Basically, we have many different viruses and bacteria can trigger the disease, and it's really unclear when a patient presents to the clinic which virus or bacteria did trigger it. So ME/CFS as an autoimmune disease. As we've said, it may be considered a flu-like illness triggered by virus infection, which is like many other autoimmune diseases. Multiple sclerosis is also involves in its pathogenesis Epstein-Barr virus infection and other viruses too. Rheumatoid arthritis, EBV, is a big candidate trigger for that, etc., etc. So we've said it's immune-mediated. That is like autoimmune diseases by definition. ME/CFS has striking similarity to MS and other autoimmune diseases, except the big difference, which has created a nightmare for patients, is that there's no actual sign or pathological entity which is reproducible in every patient. So ME/CFS has subtypes like many autoimmune diseases. And as we know, multiple sclerosis has several different subtypes. Rheumatoid arthritis has 4 or 5. And generally, all the autoimmune diseases are characterized by the appearance of subtypes. And there are several examples in which different subtypes of autoimmune diseases must be treated with different treatments. So these are significant differences which it is necessary to recognize before we can actually get a clinical response from a treatment. So the abnormalities that you find in ME/CFS are also found in other autoimmune diseases, different autoantibodies, etc., different alterations in the profile of immunity. So exhibits various autoantibodies, and ME/CFS exhibits a lot of comorbidity with other autoimmune diseases, such as fibromyalgia, POTS, Hashimoto's disease, and a family history of autoimmune disease. Some CFS patients may benefit from rituximab, which is B-cell depletion, and this is the case with other autoimmune diseases. It's not all patients who respond to that treatment. Again, the existence of subtypes. So ME/CFS exhibits upregulated Epstein-Barr virus-induced gene number 2, which is a human gene which is induced by EBV infection. And this is also true, back one, please. And this is also true of other autoimmune diseases, such as multiple sclerosis. Studies have shown upregulation of EBI2, other autoimmune diseases as well, type 1 diabetes, rheumatoid arthritis, and various cancers. So we're going to talk more about this gene EBI2 later, because it's one of the ones that we found in our study that was published exactly 10 years ago. Next. So this slide summarizes some microbial triggers of ME/CFS. And we have here viruses and bacteria and even vaccination. So enterovirus, chlamydia pneumoniae, Epstein-Barr virus, recurrent varicella zoster virus infection, recurrent human herpes virus number 6 infection, parvovirus B19 infection, hepatitis C virus, cytomegalovirus, and post-vaccination with against pneumococcus, measles, mumps, and rubella, and influenza. So basically, this is my own creation to illustrate the problem. And that is, we have multiple subtypes, probably each with a different pathogenesis, but with overlapping clinical presentations. Thank you. So basically, we've mentioned that psychological stress is important in virus infection. It's actually important in virus transmission and infection. And these were studies from the Common Cold Unit in which people were challenged with the viruses of the common cold, of which there are several, and their psychological stress index was calculated. So basically this consists of perceived stress, negative life events, and other aspects like that. And it's a composite score of the amount of psychological stress that that person was under. And you can see here, when the psychological stress rises, the number of patients with symptomatic colds rises too, upon challenge. And this is also the case when we look at different strains of the viruses causing the common cold. Next, please. So this is my own study on psychological stress in parvovirus B19 associated ME/CFS. And here again we have the stress index with the patients with fatigue. And in this study, it was a small study, and we had patients with prolonged fatigue, which was 3 months or less, and we had patients with ME/CFS, which was 6 months or more. 3 months to 6 months for prolonged fatigue, and ME/CFS greater than 6 months. Because in this study we used the Fukuda criteria. And you can see again that for acute infection and follow-up, the acute infection was the existence of fatigue at all, and the follow-up were those two categories, the prolonged and the ME/CFS. That basically raising the stress level made fatigue more likely. In Epstein-Barr virus infection, this is well recognized. So psychological stress has a variety of effects on the immune system. And it reduces natural killer cell activity. It reduces interferon gamma production from cells of the immune system in general, reduced interleukin-2 receptor expression, reduced proliferation in responses to mitogens, reduced T-cell proliferation to Epstein-Barr virus peptides, reduced T-cell killing of EBV-transformed B cells, reduced antibody and T-cell responses to hepatitis B vaccine, and upregulation of pro-inflammatory cytokines. And all of this basically represents a weakening of the cytotoxicity of the immune system. And what that does is it allows Epstein-Barr virus to reactivate. Because controlling the virus depends on having a very effective, strong, cytotoxic arm to the immune system. And when we weaken that with psychological stress, it leads to all these abnormalities and a reactivation of virus. Next, please. So, Epstein-Barr virus. This is a large double-stranded DNA virus. It's enveloped because it buds from human cells and takes the cell membrane with it. It infects B lymphocytes and possibly epithelial cells, and it transforms these B lymphocytes. And what that means is normally cells, depending on where they are from in the body, they have a capacity to divide and regenerate. And transformation is the process by which a cell is allowed to keep on dividing forever. And that is a necessary step in development of cancer. And Epstein-Barr virus does cause a lot of different cancers. Next, please. So basically what you've got with Epstein-Barr virus is two different phases of infection. A latent infection which exhibits reduced transcription, no replication of the virus, so new viruses aren't produced. And this latent phase is necessary for the virus to persist, which it does for the rest of our lives. And 90 to 100% of people in this room have had EBV infection, and it remains in our bodies for the rest of our lives. So lytic infection is the name given to a reactivated virus. And in this case, there's unrestricted transcription. There's replication of the virus to produce infectious virus. And you can then express that virus in your fluids, and it is possible to transmit that virus. Next, please. I think I'll have a drink of water. So the transmission and epidemiology of EBV, it's transmitted by saliva, and this can be through kissing, and it's usually not just, it would need some exchange of saliva, so it's not just a little kiss, more involved sexual type kiss. It can also be acquired from a contaminated environment, contaminated by saliva containing the virus. And the epidemiology depends on the environment. So classically, it's thought that the transmission is different, and epidemiology, between the developing and the developed world. For example, in the developed world, we have better standards of hygiene, smaller families, and so the transmission of the virus generally doesn't happen until we are teenagers. At this time, we meet members of the opposite sex and kiss them, and we transmit the virus that way. So at age 16 to 18 to 20 or so. But in the developing world, where generally there are larger families, maybe lower standards of hygiene, the virus is transmitted in infancy or as young children. And again, via saliva. So 90% to 100% of adults are seropositive worldwide. And as we've said, the virus persists long term. And this was a graph showing the seroprevalence with age, and you can imagine that it goes down a bit below. But basically it rises up to 90, 95, 100%. Next, please. So the pathogenesis of the virus infection in an immunocompetent host, because that's quite important, is that you acquire virus from saliva. There's an incubation period of 2 to 7 weeks. In childhood, it tends to be asymptomatic, almost all the cases. But at the time of teenage years, 30 to 50% develop infectious mononucleosis. We'll go through the symptoms of that. But basically, primarily, it's a B lymphocyte infection. And within the first 2 weeks, there's a whole army of immune response coming to control that. And these are the cytotoxic immune system components. Next, please. So these are the symptoms of infectious mononucleosis. Principally, we have fatigue, fever, sore throat, lymphadenopathy, rash, and in some cases, enlarged liver and enlarged spleen. So obviously, this is remarkably similar to the disease that ME/CFS patients suffer. Next, please. So associations between Epstein-Barr virus and autoimmune disease, there are many, but these are the highlights. Multiple sclerosis, which has a lot of symptomatology overlapping with CFS, ME/CFS. Type 1 diabetes mellitus, rheumatoid arthritis, systemic lupus erythematosus, and of course, ME/CFS. EBV is also associated with a variety of different malignancies. Burkitt's lymphoma, Hodgkin's lymphoma, stomach cancer, nasopharyngeal carcinoma, and post-transplant lymphoproliferative disorder. And basically, the virus has to be on board for many years before producing these manifestations. Next, please. So, how does EBV reactivation occur? Under what circumstances can it cause psychological stress leading to ME/CFS? Well, different examples are, studies have shown medical students at exam times under a lot of stress, military recruits at exam times, reduction in markers of cellular immunity, as we've discussed, there are many, and this all results in the reactivation of the virus to cause a clinical infection, which can be many years distant to the time of acute infection. And this is also seen with other herpes viruses. They characteristically reactivate under stress, such as the herpes virus-induced cold sores, varicella zoster virus. Once it infects, it can hide in the different nerve ganglia and reactivate to produce disease under times of psychological stress. Next, please. So what are the types of stress that can weaken the cellular immunity? Well, marital stress. I'm sure those who are married know all about that. Student examination stress, attachment anxiety, fear of abandonment or rejection, loneliness, but not physical stress. So these are psychological situations which create immense fear, and we react over time by this having a bad effect on our cytotoxic immunity. Slide, please. So how does this happen? What's the pathogenesis of this mechanism? So basically, when you have road rage or whatever, or when you fight with your husband or wife, basically that causes psychological stress, which leads to release from the adrenal cortex of glucocorticoids. And these are steroids which help us cope under times of stress. But these lead to expression. These allow the expression of Epstein-Barr virus immediate early gene BZLF1. So that's the first gene to be expressed in a reactivation. And then Epstein-Barr virus encoded DUTPase upregulates NF-kappa B expression. And what this means is that this product of the virus is able to interact with the DNA. And there are these NF-kappa B sites all throughout our genome, and they basically the binding of that alters the immune response so that raised levels of pro-inflammatory cytokines are produced. And this is a so-called Th2 pattern of T helper cell immune responsiveness. And it's not good when you want to control Epstein-Barr virus. So basically this DUTPase reduces innate immunity in monocytes, and it induces sickness response in mice. So sickness response is the symptoms of ME/CFS. It's being tired. If we do too much, whether we have ME/CFS or another autoimmune disease, or whether we're normal, if we do too much, we experience a sickness response. And in that case, we want to go to bed early. We feel tired. We feel we've got pain. We might feel depressed for a time or a bit negative, and we go to bed early and sleep well, hopefully, and feel better in the morning, if we're lucky. So restraint stress impairs learning and memory due to Epstein-Barr virus DUTPase production. And restraint stress is mimicked in the lab by putting a mouse inside a small tube, and it can't really move. It can breathe, but it can't move, and there's a struggle, and that is restraint stress, and that is a form of psychological stress. Slide, please. Okay, so now we're going to talk about the EBI2 gene, and this is important in immunity and brain function. So I'll show you this slide shortly, showing that we identified this gene as being upregulated in ME/CFS. And at the time, it was very interesting, but I didn't understand what it meant. And I spoke to various people about it, and nobody had any ideas. It was a gene that was known to be induced by Epstein-Barr virus infection of lymphocytes, but the function wasn't known and not much was known about it. So it was published and we didn't do any more. But in the last 10 years, there's been an enormous amount of work on this gene, and it's been shown to be important in immunity and brain function. So what is it? It's a G protein coupled receptor. So it's a human gene which spans the cell membrane from outside to inside, and involves these G proteins, which are subunits of this type of receptor. They're just called G proteins, and there are a number of them, and they get together to form the receptor, and it's a signaling mechanism within the cell. So this is the main induced gene in Epstein-Barr virus-infected Burkitt's lymphoma cells, and it's upregulated 200 times. It's highly expressed in the blood cells, peripheral blood mononuclear cells, sorry. In all of them, actually, but principally the lymphocytes and the NK cells. It's a regulator for B cell partitioning in lymphoid tissues. So the B cells must go through various stages of development in the lymphoid tissue before they emerge as antibody-producing cells or plasma cells. And their journey through their stages of development is very much dependent on EBI2. It's critical for T-cell mediated antibody responses. It's a negative regulator of the innate immune response, and we know that ME/CFS patients exhibit defects in the innate immune response, particularly with NK cell function. It's upregulated in various autoimmune diseases. Its precise role in EBV infection remains unknown, however. And oxysterols are the ligand, so-called ligand, for the receptor. So basically, any receptor must have a ligand, and what that means is it's the other part, the connection in the signaling pathway. So the G protein coupled receptor is at the cell membrane, and these oxysterols come and bind that to begin the process. Slide, please. Oh, well, this was our gene expression study of 10 years ago, and somewhere out of sight you'll see that gene. But anyway, we found that it was significantly upregulated in ME/CFS patients as compared with normal controls. And that was in a microarray experiment and with individually tested genes by reverse transcriptase PCR. Slide, please. And this is just to show that these patients were, the genes we identified, the 88 genes, we clustered them, which is an undirected process allowing the genes to settle out into groupings which we assume means something biologically. And when we did that, we obtained 7 different clusters, and then we looked at the patients in each of the clusters for their clinical details, and this here is the SF36 score, the different domains of that and the total. And you can see that these 7 different subtypes were significantly different from each other. Slide, please. So, EBI2 in ME/CFS. It appears to be upregulated in approximately 38 to 55% of ME/CFS patients. Its upregulation may be associated with severity, and I'll present a little data on that. EBI2 upregulation is associated with upregulation of other EBV-associated genes and proteins. And the highest EBI2 levels were associated with low level of anti-EBNA IgG. And this was also found by a study, this study here, in which low levels of anti-EBNA IgG were found in ME/CFS patients. So EBNA1 is an Epstein-Barr nuclear antigen, as a multifunctional protein necessary for multiple functions. Therefore, low anti-EBNA IgG may be associated with EBV reactivation. Slide, please. Okay, so this is our data. What I showed you there was the 88 genes in ME/CFS patients versus normal. the different clinical syndromes in each of the subtypes. And this slide focuses on the comparison between ME/CFS patients and normal controls for EBI2 and NTE. So EBI2 is the gene we're discussing. NTE is neuropathy target esterase, which does have a very important role to play in people exposed to organophosphate pesticides and chemicals, which, as we know, have been shown to trigger ME/CFS. So basically this little pie, open pie symbol is the EBI2, and this star is the other gene shown here, NTE. And you can see that in approximately 50% of patients, there are upregulations of EBI2. In the normals, nobody has a raised level. There are a few here, but basically the normals show a uniform low level, whereas the ME/CFS patients show significant variability. So some patients have upregulation and others do not. In the case of NTE, the other gene, 2 patients have upregulation. Slide, please. Now, this study was different, and in this case, we took 117 ME/CFS patients, the gene expression data from these, clustered it, and in this case, because there were more patients, we identified 8 different subtypes. And this again was published in 2008. So you can see the SF36 score here. The more individual symptoms are shown in this slide. And these patients, these clinical syndromes were really quite different from each other, some more than others. Over here we have the different antibody markers of the disease, and this one here is the EBNA IgG, and we have a low level in subtype D, which was associated with greater severity, and one of those patients, actually their disease was triggered by a documented, laboratory documented EBV infection. And these were the genes, the other genes in the study, which we found to significantly vary with the level of, with the different subtypes. And you can see here that the levels of these genes were very high in subtype D compared with subtype C, for example. Slide, please. So what are the markers of Epstein-Barr virus reactivation? Because if we did have a treatment, then we would need to know in which patients we should be using it. So these are those that are in the literature so far. IgM antibodies to viral capsid antigen, to early antigen, to the EBV DUTPase, and the DNA polymerase. These 2 antibodies to this were first published upon by Michael Lerner. IgM to early antigen is another marker, and VCA as well. IgG to early antigen, DUTPase and DNA polymerase. Again, these 2 first were discovered by Michael Lerner. And ELISpot test, and this is an enzyme-linked immunospot, testing the reactivity of cells, immune cells, to different EBV proteins, those that are latent and those that are lytic. So basically it gives a snapshot of what the immune system is doing regarding this virus at that moment. And if you have high reactivity to both latent and lytic, well, obviously you have a reactivation. Otherwise, there wouldn't be the lytic reactivity. And finally, I believe that our gene EBI2 is a marker of EBV reactivation as well. So we have a number of them here. Slide, please. So in ME/CFS patients with reactivation, what are the options for treatment? Well, there's two, valacyclovir, and more specifically for EBV is valganciclovir. The problem with valganciclovir is that it's very expensive. EBI2 antagonists are in development, and but these are also likely to be very expensive. And the final one there, cimetidine, was first developed as an H2 receptor blocker to treat acid reflux in the stomach. But it has this really intriguing side effect that it inhibits Th1 suppressor cells. And those cells are responsible for reducing the cytotoxicity of the immune system. And so when you inhibit them, you get an enhanced cytotoxic response in your immune system. So,
sorry, would you give that with the antiviral? Would that be part of a package of a protocol for something?
well, we can.
Would you give it as opposed to the antiviral? Would you give it together? Or?
Well, there's been no trials of this, but the only thing, so it's difficult to answer the question, but Jay Goldstein published 2 letters, treatment of chronic Epstein-Barr virus disease with H2 blockers. This was in 1986. And then cimetidine, ranitidine, and Epstein-Barr virus infection. And this again was in 1986. And basically, he was treating his patients who he believed had EBV reactivation. Some of these had ME/CFS with cimetidine. And he states in these letters that there was a beneficial clinical effect. And the rationale for that, I've explained to you, is that you enhance the cytotoxicity of a person who's taking this drug. The other interesting thing about cimetidine is if it was going to be useful at raising the cytotoxic response, then you would expect that the virus suppressed cancer as well. And many of these people have been on cimetidine for several decades, and there are several epidemiological studies have shown that in patients who take cimetidine for a long period of time, there is a significantly reduced incidence of a variety of cancers, and that is very much in support of its use for treatment of Epstein-Barr virus reactivation. Slide, please. So, thank you for your attention.
Anybody any questions for Dr. Kerr? Just put your hand up and we'll take them as they come. Come on, don't be shy.
Doctor, you see, the other way, a tick bite and they say it causes Lyme disease. Is that any connection to, what I was bit by ticks as a child?
Does Lyme disease kind of...
Does it stay in your system or does it clear?
It gets cleared,
Right.
but it can cause a very multi-system illness and a lot of fatigue, yeah.
Yeah, well, I have arthritis, 2 types of arthritis, but I think it's in the gene through the mum's side.
Anybody else any questions?
I have a daughter last year with full-blown reactivation of EBV, and she's been on 18 months of valacyclovir,
Okay,
and she has regained 50% of the previous functionality.
okay.
She has had ME for a long time. We don't use ranitidine at the minute. In your opinion, obviously, Dr. Montoya recommends obviously longer-term use of it, and in line with what Dr. Nairn was doing as well. What can be done to strengthen or improve the immune function, the NK function? And what's
Well,
the role of CD8 in terms of...
CD8. Well, Jay Goldstein